Next, we are going to discuss about the carbon and energy sources for the bacterial growth. In order to grow in nature or in the laboratory, a bacterium must have an energy source, a source of carbon and other required nutrients, and a permissive range of physical conditions such as oxygen concentration, temperature, and pH. Sometimes the bacteria are referred to as individuals or groups based on their pattern of growth under various chemical or physical conditions. For example, phototrophs are organisms that use light as an energy source. Then coming is anaerobes. Anaerobes are the organisms that grow without oxygen. Then thermophils. Thermophils are the organisms that grow at high temperatures. Next coming is about the growth factors which are required for the growth of the bacteria. The simplified scheme for the use of carbon, that is either organic carbon or carbon dioxide, ignores the possibility that an organism, whether it is an autotroph or a heterotroph, it may require a small amount of certain organic compounds for their growth, because these organic compounds are essential substances that the organism is unable to synthesize from the available nutrients, and such compounds which the organisms are unable to synthesize from the available nutrients are called growth factors. The growth factors are required in small amounts by cells because they fulfill specific roles in biosynthesis. The need for the growth factor results from either a blocked or a missing metabolic pathway in the cell. At that time, growth factors are needed. The growth factors can be organized into three categories. First one, purines and pyrimidines, which are required for the nucleic acid synthesis, such as DNA and RNA. Then next is amino acids. They are also required for the synthesis of proteins. Next coming is vitamins. They are needed as coenzymes and functional groups of certain enzymes. Some bacteria, for example E. coli, do not require any growth factors. They can synthesize all the essential purines, pyrimidines, amino acids and vitamins starting with their carbon source as the part of their intermediary metabolism. There are certain other bacteria, for example lactobacillus. They require purines, pyrimidines, vitamins and several amino acids in order to grow. These compounds must be added in advance to culture media that are used to grow these bacteria. The growth factors are not metabolized directly as sources of carbon or energy. Rather, they are assimilated by cells to fulfill their specific role in metabolism. Mutant strains of bacteria that require some growth factors and which are not required or needed by the wild type strains. They are referred to as auxotrophs. So, the mutant strains requires growth factor, whereas wild type strains which doesn't undergo any mutation, they doesn't need these growth factors. Thus, a strain of E. coli that requires the amino acid tryptophan in order to grow would be called tryptophan auxotroph and would be designated as E. coli TRP because they require the amino acid tryptophan because they have undergone some types of mutation. So they are called auxotrophs. There are certain vitamins which are frequently required by the certain growth factors as in the bacteria, and the function of these vitamins in essential enzymatic reactions gives a clue why, if the cell cannot make the vitamin, it must be provided exogenously in order for the growth to occur. So for the occurring good growth, it should be provided exogenously. Which one? Vitamins. Next are the common vitamins required in the nutrition of the bacteria. First vitamin, para-aminobenzoic acid. That the function is it acts as a precursor for the biosynthesis of folic acid. Next is folic acid. And the coenzyme of this folic acid is tetrahydrofolate. The function is it transfers one carbon unit and required for the synthesis of thiamine, purine bases, serine, methionine and pantothenate. Next vitamin is biotin. The coenzyme for the biotin is biotin itself. The function of this biotin is biosynthetic reactions that require carbon dioxide fixation. Next is lipoic acid. The coenzyme of this lipoic acid is lipoamide. Function, it acts in the transfer of acyl groups in oxidation of keto acids. Next is mercaptoethane sulfonic acid. It's a mercaptoethane sulfonic acid. The coenzyme is coenzyme N. The function is methane production by methanogens. Next vitamin is nicotinic acid. The coenzyme in the nicotinic acid is nicotinamide, adenine dinucleotide and NADP. Functions, it acts as an electron carrier in dehydrogenation reactions. Next is pantothenic acid. Coenzyme required is coenzyme A and the acyl carrier protein. Function, oxidation of keto acids and acyl group carriers in metabolism. Next is piridoxin. Coenzyme is pyridoxal phosphate. Function, acts in the transamination, deamination, decarboxylation and racemation of amino acids. These are the function of the B6 vitamin, that is piridoxin. Next is riboflavin, vitamin B2. The coenzyme is FMN, which is flavin mononucleotide, and FAD, flavin adenine dinucleotide. These two are the coenzymes of riboflavin. Function, it acts in the oxidoreduction reaction. Next vitamin is thiamine, which is also known as vitamin B1. The coenzyme used are thiamine pyrophosphate. The function is decarboxylation of keto acids and transaminase reactions. Next vitamin is vitamin B12, which is also known as coenzyme, that is cobalamin coupled to vitamin B12 is what? Vitamin B12, the name, that vitamin, it acts as an intrinsic factor. The vitamin B12, the coenzyme of this vitamin B12 is cobalamin coupled to adenine nucleoside. The function of this vitamin B12 is transfer of methyl groups. Next vitamin acting as a growth factor is vitamin K. The coenzyme used in this vitamins are quinones and naphthoquinones. Then the function of this vitamin K is in the electron transport process. So these are all about the nutritional requirements of bacteria and also the growth factors. In the next session we can discuss the classification of the bacteria based on nutrition. Thank you.